The next thing we want to touch on is transcription, which is the method whereby information in the DNA, which contains instructions for building a particular protein, is copied to a novel molecule called a messenger RNA molecule. When this happens, essentially what the cell is doing is requesting a certain gene product be produced, and in order to do that, what has to happen is that transcription factors have to enter the nucleus through nuclear pores and then search out the promoter, which is the front part of the gene, and then through interaction with that promoter, unwind that section of DNA so that an enzyme called RNA polymerase can then copy that information on the DNA into a novel polynucleotide called a messenger RNA molecule. So the RNA polymerase will catalyze the formation of the messenger RNA using the information in the DNA strand as a template and following the rules of complementary base pairing. Essentially, anywhere we see an A in the DNA strand, we're going to incorporate a U. Anywhere we see a C in the DNA strand, we're going to incorporate a G. And what will happen as a result is that you'll get a perfect complement of the strand of DNA known as the coding strand. It'll end at a sequence of DNA known as the terminator sequence, which contains sequence information that indicates to the RNA polymerase that it no longer needs to continue to incorporate nucleotides into the message, and the result is that the RNA will disengage from the DNA strand. Once we've made enough message off of that particular gene, what will happen is that the transcription factors will disengage from the promoter, and that section of DNA will wind back up until the cell needs that particular gene product again. Remember that when we're talking about gene expression, it's entirely possible to make more than one copy of a transcript off of a single gene if that cell needs that product in particular abundance. And in addition to that, once that message is processed, several things have to happen before that message ends up being translated by the ribosomes in the cytoplasm. It has to be spliced, meaning that sequences that don't contain protein coding information have to be chopped out of the transcript. The exons have to be then sealed together, and then the message has to be polyadenylated at its 3' end and capped at its 5' end. And once that happens, it's ready for translation once it leaves the nucleus through the nuclear pore. You can make more than a single copy of a transcript for a particular gene, and in addition to that, when we're translating that message, we can translate it more than one time to make sufficient protein to satisfy the cell's needs. So you have to remember that there's the potential for making a whole lot of gene product here if it's warranted by the needs of the cell. So this is a look at the stages of transcription.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter and a terminator downstream of the coding region. The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase attaches to the promoter region and forms an open complex. RNA nucleotides are then polymerized from nucleotide triphosphates to make a messenger RNA. The messenger RNA sequence is elongated as the RNA polymerase moves down the DNA molecule until the RNA polymerase reaches the terminator region. Transcription is terminated when sequences in the terminator region are encountered. This causes RNA polymerase to dissociate from the DNA molecule, and the completed transcript is released.
Now, post-transcriptional processing is important because in the DNA, there are regions of sequence, regions of nucleotides that don't contain information that's going to end up contributing to the structural information that goes into making the protein. And for the longest time, it was thought that these regions of DNA simply had no purpose, but it turns out that several decades after their discovery, we've determined that it's these introns that probably contain regions of DNA that control when and where a gene is expressed as a result of binding to types of transcription factors, gene modulators, and also, it turns out, through the production of RNAs that aren't strictly speaking coding for a protein, but are really there to serve to modulate the rate of transcription or to modulate the rate of translation of a particular message. So these introns contain a lot of control sequences, and that's why they're simply not useless chunks of DNA. We don't see introns in prokaryotic cells like in bacteria. Every piece of information in the DNA is represented in the message, and in fact, in prokaryotes, it turns out that transcription and translation happen simultaneously, and the reason for that is that in prokaryotes we don't have a membrane-bound nucleus. The genetic material is actually in the cytoplasm. So as the RNA polymerase is transcribing the gene, the ribosomes that are in the cytoplasm are at the same time translating it. But in eukaryotes, it's a very different sequence of events because we have the nuclear material in a compartment in the nucleus, which is bound by a double-layered membrane that contains, of course, nuclear pores. So we have to transcribe that information into a messenger RNA first. The next thing that has to happen is that any intron sequences have to be spliced out. The exons are then joined to each other. Next thing that will happen is that the message will be capped by a very special nucleotide called 7-methylguanosine, and this indicates basically where the front of the message is, and it helps stabilize the message. And then a poly A tail is added to the 3' end, and we think that the poly A tail may have a role both in gene regulation and may also have something to do with how long that particular message hangs out in the cytoplasm or attached to the rough endoplasmic reticulum. So some of the thinking is along the lines that the longer the poly A tail is, the longer the message will hang around. The shorter it is, the more likely the message is to eventually be broken down and that gene product no longer be deemed necessary by the cell. And so this is something that we see in most eukaryotic genes. Now, there are some eukaryotic genes that don't have introns. Okay, there are some eukaryotic genes that have a single exon. There are some eukaryotic genes that have many exons and many introns, and there are some eukaryotic genes that exhibit something known as alternative splicing, where we'll have certain exons that end up in the message in a particular flavor of that messenger RNA, and then other exons that end up in the message in another flavor of that particular messenger RNA. So this gene expression is a lot more complex than simply opening up the DNA and copying the information and then sending it out of the nucleus to be translated. So let's talk about translation. This happens on ribosomes, and what they do is they read the information on the messenger RNA and use that to select transfer RNAs that are attached to the appropriate amino acid and incorporate those into the growing polypeptide chain. The RNA species involved in translation are ribosomal RNA, which is a structural component of ribosomes, transfer RNA, which of course is the adapter molecule, which is connected at one end to a particular amino acid, and there is another structural feature called the anticodon loop, which is what the ribosome actually uses to pair up that transfer RNA with the codon that's in the message. And then finally, we have the messenger RNA, which by this time contains the information needed by the ribosome to properly translate the sequence of nucleotides into a sequence of amino acids, which will be the protein. Now, there are some parts of the message which are what we call non-coding, okay? There are what we call 5' untranslated and 3' untranslated regions, and those are there, again, primarily to regulate translation. So sometimes there will be regulatory elements that bind to these untranslated regions that can either permit or deny the ribosome's entry into the coding part of the message. Sometimes they don't have a regulatory function. It all depends on that particular gene. But what will happen when translation begins is that the ribosome will bind the 5' end of the message and then scan for the initiating codon, which in most eukaryotic genes is an AUG. And if it finds an AUG in the proper context, meaning with the proper sequences the 5' and 3' end of that particular start codon, what it'll do is it'll pause, and then it'll begin the process of translation. There might be in a 5' untranslated region of a gene several AUGs, but only one will represent the beginning of the reading frame, which corresponds to the protein that the cell needs produced. So the transfer RNA anticodons will match with the messenger RNA codons. The ribosomal RNA catalyzes the formation of a peptide bond between the amino acids. Remember that those occur between the carboxylic acid group of one amino acid and the amino group of the amino acid that follows it. So it's a very particular type of covalent bond. And what will happen is we'll begin to slowly incorporate amino acids running now from the carboxy terminus to the amino terminus of the amino acid. Basically what happens when the first messenger RNA is brought in is that the amino end is going to represent the first amino acid in the chain, and the carboxyl end is going to end up representing the last amino acid in the chain. And so what happens is that we proceed 3 bases at a time from the beginning to the end of the message, and we incorporate now the appropriate transfer RNA that contains the codon that's going to be a match to the anticodon. They're going to show complementarity. That's how the ribosome knows that it's selected the right molecule, and it'll move down 3 bases at a time until it reaches the end of the message, which will contain a terminator codon, which will code for a transfer RNA that's not attached to an amino acid. Instead, it's attached to a water molecule. And once that's incorporated into the polypeptide chain, it'll be released. The ribosome will fall apart, and then that may be the end of the story. That may be the end of the production of that particular gene product. That gene product will be released into the rough endoplasmic reticulum in the case of a ribosome that's bound to the rough ER, or it'll be released into the cytoplasm if it's being translated by the cytoplasmic ribosome. Or it may be the case that many ribosomes will march down this particular message just like cars on a train, each producing a single copy of the polypeptide chain encoded for by that particular message. And what you can see here is just how it is we incorporate the amino acids in the growing chain. You can see how the ribosome binds and scans for the initiating codon. We then bring in the first transfer RNA, which is bound to an amino acid called methionine, and then we bring in the second transfer RNA, which is going to have an anticodon complementary to the second codon in the reading frame. Will then catalyze the formation of a peptide bond between amino acid number 1 and amino acid number 2. The ribosome will then slide down another 3 bases. It will kick out the first transfer RNA molecule, which is now no longer linked to an amino acid. It will head off into the cytoplasm where other enzymes will recharge it so that it can serve in future translations. And then we'll be sitting now on the top of the third codon. We'll bring in now the third transfer RNA. The process will continue to repeat until we get all the way down to the terminator codon, at which point, of course, the ribosome will split apart, the gene product will be released, and we may end up having that be the end of translation, or, as pictured here, you may have several ribosomes marching from the 5' to the 3' end of the message, translating it. One of the things that we have to understand is that when we see more than one ribosome translating that particular message, that's a phenomenon known as a polysome. Okay, it means polyribosome. And so what you're seeing in here is a need by the cell for a lot of this particular gene product in order to fulfill a particular need for the cell. So this is just a look at how protein synthesis operates.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of one codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site and the ribosome shifts down by a distance of one codon. This is followed by the two amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
Okay, all nucleated cells except for germ cells have a full complement of DNA. Remember that germ cells contain half the amount of genetic material as a normal body cell does. So in a human being, a sperm or an egg cell will contain 23 chromosomes, while a body cell will contain 46. The 46 is the diploid number, the 23 would be the haploid number. During development, differentiation occurs, and what happens is that after fertilization, what will occur is the zygote, which is the fertilized egg, will begin to divide very rapidly. And what will happen over the next several weeks is that cells that are destined to be particular tissue types, say muscle cells or nerve cells or cells that become part of the skeletal system or cartilage, will gradually be restricted in terms of which DNA they're allowed to read in order to direct their fate. So while all the genetic material is the same in every body cell, what makes the cells different is the different genes they're allowed to use to guide their actions, their appearance, and their development. So after a time, as an example, a cell might go from being essentially an undifferentiated cell that could be anything to gradually looking more and more like a muscle cell. And what guides that is that genes that aren't important in muscle cell function, while they'll still be present in the nucleus, will no longer be available chemically. They'll essentially be sealed off from transcription factors so that they can't be used to guide the behavior of that particular cell. And so what will happen is that the cell will become more and more restricted in terms of what its fate can be, because it will now be reading from an ever shrinking amount of information in the nucleus. During the lifetime of the cell, the rate of protein synthesis will vary depending on chemical signals that reach that cell. As an example, thyroxine from the thyroid gland, which is a butterfly-shaped gland right in front of your neck, can cause cells to increase their metabolic rate. That's essentially the rate at which they convert macronutrients into readily available fuel in the form of ATP. It also accelerates the amount of heat that's produced. The more thyroxine, the higher the metabolic rate. The less thyroxine, the lower the metabolic rate. That's an example of a cell encountering a signal in the external environment, which, as a result of interaction with receptors, and in the case of the hormone thyroxine, the receptor is actually inside the cell. The receptors binding to the ligand produce a conformational change that ends up activating, in the case of thyroxine, particular genes in the nucleus. In the case of hormones that can't cross the plasma membrane and instead have their receptors on the membrane surface facing the extracellular environment, conformational changes in those receptors set off a cascade of enzyme activation in cytoplasmic molecules that ends up eventually either turning on a particular enzyme that makes the cell perform a given action, or ends up sending an activated protein like a transcription factor into the nucleus where a particular gene will be turned on or turned off. And so that's going on all the time. So this is sort of the cell membrane's role in terms of monitoring what's going on in the outside world, looking for information, dictating what it's supposed to do. Now, the cell will approach a point in its lifespan where it's going to be time for it to go from being one cell to being two. Most of the cell's time in most tissues in the body is spent in interphase, where the cell is essentially performing its task, being part of the organism. But at some point, what will happen is that there will be outside signals that will affect processes inside the cell that tell it to reproduce its genetic material, so that we go from essentially a 2n number to a 4n number. Okay, so we go from being diploid to, right before mitosis begins, actually being tetraploid. And what will happen is that that information, once it's been duplicated, will be condensed into visibly staining bodies known as chromosomes. Those chromosomes, which have been duplicated in the course of mitosis, will then be pulled apart, and then each new cell will get a complete copy of the genetic material. So what you'll end up at the end of mitosis is two new diploid cells, each with the exact same amount of genetic material as the original parent. So that's the series of events that leads to the production of two cells by the division of the mother cell into the daughter cells, and the cells that are produced are going to be genetically identical. And the reason that that's important is because the two new cells, since they're using the same DNA, there's a reasonable expectation that they're going to look and act the same as the original parent cell. So the phases of mitosis include prophase, metaphase, anaphase, and telophase. The way to remember that is PMAT. Just keep that acronym in your head. And then generally following that, in 99% of the cases, the rest of the cell contents will divide, and that's a process known as cytokinesis, where the organelles, the cytoplasm, the cell membrane end up being partitioned generally equally between the two new daughter cells. There are some exceptions to that. So let's take a look at how the cell cycle operates.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing two copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has four stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the two groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, The chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of two genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of four phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of two genetically identical daughter cells.
Okay, now, when the cell gets ready to reproduce its DNA, the strands have to separate. The old strands will become templates for the new strands to form. And this process is known as semi-conservative replication. What's unique about the structure of DNA is the fact that because of its double-strandedness and because of the fact that the strands are complementary to each other in addition to being anti-parallel, when the DNA double helix unravels, each old strand contains the information in it that can be used by the enzymes that make new DNA, which are called DNA polymerases, to make the new strand. And that was really the huge breakthrough that came about as a result of elucidating the structure of the DNA molecule. This is a look at how this operates. You can see here that at very special spots along the DNA molecule called origins of replication, what will happen is that the double helix will open up. And as the double helix opens up, proteins whose job it is to stabilize the separated double helix will move in, followed by proteins whose job it is to copy the new strand using the information that's in the old strand. And so what you can see here in this diagram, the new strand is shown here in pink, okay? The old strand here shown in blue, okay? So this is what we mean by semi-conservative replication. When the DNA finishes being duplicated, each new DNA molecule will contain a strand of information from the original DNA molecule and also a strand of newly synthesized DNA. And again, this generally works pretty well. Usually we get a true copy of the DNA molecule, which ends up being distributed to the two brand new cells. Occasionally what can happen in this process, though, is that a mistake can be made. We can, as a result of copying this template, goof up and put a wrong nucleotide in the new strand. Fortunately, there are enzymes in the nucleus whose job it is to look for those mistakes and fix them. They're called DNA repair enzymes. But sometimes what will happen is that that mistake won't get caught. And if that occurs, that results in what we call a mutation. And this happens every day in everybody in class. You generally have mutations that occur in different tissues all the time. Now, if these mutations occur in genes that are critical for the function of the cell, what can happen is it can change the way the cell looks and acts. It could cause the cell to die, or in another scenario, it could cause the cell to begin to divide in an uncontrolled fashion, and this is what we call cancer. Fortunately, we have another fail-safe built in to guard against these occasional changes that take place as a result of mistakes in the replication machinery, and that's our immune system. Our immune system's job is to keep us disease-free. Its other job is to scan the tissues of the body and look at their antigens, which are collections of molecules on the membrane surface that identify that cell as belonging to that organism. And if they find changes in those antigens, what the immune system will do is target those cells for destruction, because now they're recognized as not belonging to the organism anymore. So wipe them out before they get a chance to spread. Occasionally what will happen is that we'll get a changed cell that may be identified by the immune system, but may be able to escape being destroyed as a result of its own chemical tricks. And there's a variety of strategies that cancer cells can use to avoid being destroyed by the immune system. One is to destroy the immune cells that come to kill them. The other is to try to disguise themselves so that they look enough like ordinary cells that they're not picked up by the immune system. There's any number of strategies that have been described. In the event that the immune system does not identify these cells and destroy them, they can become malignant and then spread throughout the body. It's one of the unusual properties of neoplastic cells that they can move from their original source of generation, where the original mutation took place. They can get into the circulatory system or the lymphatic system, and they can spread from there throughout the entire body. And then wherever they land, they can set up a new tumor mass. The reason that this is a concern is because these cells are rapidly dividing and they're not really providing any benefit to the host. Instead, what they're doing is hijacking oxygen and nutrients from the circulatory system at the expense of the organism's existence. And the result, after a time, is that the tumor cells become so highly populated, so widely dispersed, that the organism can no longer survive, and that ends up being lights out for the individual. So this is why we take such great precautions to identify these types of changes as early as we possibly can. And there's a variety of techniques that we can use to do that. Some are immunological in origin. Some rely on a technique called PCR. Essentially, if we detect cancer cells when their numbers are low, the treatments that we have to deal with this are going to be more effective because we're more likely to knock out the cancer cells when they're very low in number than if we wait until they spread throughout the entire body. And the three broad categories of therapy that are in wide use today to deal with cancer are chemotherapy, radiation therapy, and immunotherapy. Chemotherapy works by attacking rapidly dividing cells. The chemotherapeutic agents get incorporated into these cells' genetic material. They compromise the structure of that genetic material to the point where the cell can't maintain its integrity and the cell dies. The problem with that is that it not just targets cancer cells, it also targets any cell that rapidly divides. And this is why people on chemotherapy tend to have suppressed immune systems. They tend to have a low hematocrit. They lose the lining of their gut. Their hair falls out. They get very, very ill before the chemotherapy gets a chance really to knock the cancer out. It doesn't always work either. Sometimes people go on chemotherapy and it's either too late or it's just not effective against that particular cancer. One of the things that we have to understand about cancer is that these genetic changes that can result in uncontrolled cell division can occur in any gene that affects cell division, and the result is that the potential mistakes that could lead to cancer number in the hundreds of thousands, if not higher. So the other approach we have is radiation therapy. Ionizing radiation, as you know, is a mutagen. It's damaging to DNA. The idea behind radiation treatment for cancer is that we damage the DNA of rapidly dividing cells to the point where the cell can't maintain its information in the nucleus anymore and the cell dies. And it's this basically along the same lines as chemotherapy. It's just that the agent of DNA destruction is radiation as opposed to these chemicals that get incorporated into the DNA. Immunotherapy is a little bit different. In immunotherapy, what we do is we try to tag the cancer cell with an ID tag that the immune system can easily see and destroy. And there's been some success with certain types of cancers with regard to immunotherapy, a way of alerting the immune system to the fact that this neoplasm is there. And the nice thing, of course, about immunotherapy is that it's largely going to be directed at the cells that have been tagged and not against the body's healthy tissue. So it's a little bit more specific than these other types of therapies. But again, whether the therapy is going to be effective or not is in large part going to depend on the nature of this mistake in the DNA that led to the cancer in the first place. And so that's why you're probably not going to find, you're not probably not going to end up at the end of the day with a single cure for all cancers, because the mistake that leads to the uncontrolled cell division it could be in any gene at any position, and as a result could affect any number of processes. So here's a look at the structural basis of DNA replication.
DNA replication is the process in which one double-stranded DNA molecule is used to create two double-stranded molecules with identical DNA sequences. For this process to occur, the two DNA strands are separated and the DNA replication machinery is assembled at the replication fork. The two DNA strands are used as template strands to create newly synthesized DNA strands. The key feature that allows a DNA molecule to produce two DNA molecules with the same DNA sequence is the ATGC rule, also known as Chargaff's rule. According to this rule, adenine in one strand only hydrogen bonds with thymine, and guanine only hydrogen bonds with cytosine. For example, an incoming nucleotide with a cytosine base will only hydrogen bond with a guanine base in the template strand. By this mechanism, one double-stranded DNA molecule provides the information to create two double-stranded molecules with identical base sequences. Each copy contains one newly synthesized strand and one original strand. This mechanism is called semi-conservative DNA replication because half of the DNA molecule is conserved from the original molecule.
And here's a look at DNA replication, just another view with basically the same process.
Replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the two complementary strands of the DNA molecule are oriented in opposite directions and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
Okay, cell division is an important part of the life of most cells. Some cells divide very frequently, such as cells in tissues that have epithelia. Some cells don't divide very frequently, such as neurons in the central nervous system. It all, again, depends on essentially how well that particular tissue type is protected from physical damage. Rule of thumb, the more likely a cell or a tissue is to face damage, the more likely it is to have a high mitotic index, meaning it'll replace itself as the old components get worn out. In interphase, we duplicate the DNA. So we go from the diploid state to a tetraploid state right before we're ready to engage in mitosis. In mitosis, what happens is we start with prophase, the nuclear envelope will dissolve, and the DNA molecules will condense into bodies known as chromosomes. And by this point, every chromosome has been duplicated, and they'll have a particular shape to them. They'll have a central constriction, which is called a centromere, and then they'll have arms that are called chromatids. So you can see here the centromere, there's a chromatid, there's a chromatid. This is the sister chromatid of that chromatid, and this is the sister chromatid of that chromatid. Basically, this is one DNA molecule, and this is its exact duplicate. All right? And what's going to happen at the end of prophase is that the nuclear envelope will disappear, and eventually the spindle apparatus, which contains the centrioles and their associated microtubules, will attach to these central constrictions and then align these duplicated chromosomes up near approximately the center of the cell. So they'll sort of line up almost single file. At anaphase, what will happen is that through force generated by the centrioles pulling on the microtubules, you'll actually snap these duplicated chromosomes apart, and they'll begin migrating to opposite ends of the cell. And when they reach opposite ends, what will happen is that the chromosomes will decondense, the nuclear envelope will reform, and this is the portion of mitosis known as telophase, and the microtubules will begin to disappear as well. What will happen next in most cases is that the rest of the cell components will divide, and this is what's called cytokinesis. So 99 times out of 100, mitosis comes first, cytokinesis comes next. And if you think about it, that makes sense because you've got to duplicate your genetic material and divide that equally before you divide up the rest of the cell contents. If you didn't do that, what would happen if say, cytokinesis came before mitosis is that one cell might not have any genetic material and one cell might have too much. And that would adversely affect how the cell behaves in the native organism. So this is just a look at cell division. You can see here we're beginning to, we've replicated the DNA. Now prophase, you're beginning to see the chromosomes for the first time. At metaphase, they're going to line up approximately near the center of the cell. There's anaphase, there's telophase. Cytokinesis is beginning at this point, and now cytokinesis completes. And so this is a cartoon showing you these parts of the process, and this is an actual micrograph showing you a cell as it marches through the process of cell division. And generally at the end of the day, what you end up with are two cells that look and act just like the original one. Now that's not always the case. There are some unusual exceptions to that, such as in stem cell populations where the stem cell will divide and one cell will remain a stem cell while the other cell will differentiate and turn into a cell that's going to provide a particular function in the context of a tissue inside the organism that serves a particular purpose. Some cells undergo mitosis and don't undergo cytokinesis. So what they get are these gigantic nuclei that are in the center of the cell and this massive amount of cytoplasm. An example of a cell that does that is a megakaryoblast. Okay, so just keep that in mind, that there will be exceptions to this rule, but in most of the cells in the body, what we end up with after cell division are two brand new cells that are going to look and act just like the original. So here's a look at mitosis and cytokinesis. Once you watch this video, please remember to flesh out this material in your notes. Also watch the mitosis video that I've posted on Blackboard. It goes through both mitosis and meiosis, both of which are important for you to know. And I will see everybody in class. Oh, and by the way, this also marks the end of the material that's going to be covered on the first exam. Okay, so I'll see everybody in class.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between two daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into two daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.